So obviously this is a first-in-human stem cell trial. The cell product is the genesis of many investigators' preclinical science work that has looked at human interneurons and is now being carried forward by a company. So that's Neurona. So this is an industry-sponsored trial, and there's many sites across the country. And it's taken that knowledge from the preclinical science about interneuron loss in Neurona, and the preclinical colleagues that did the work that led into the formation of that company have spent a lot of time trying to understand the transcriptomics, the nature of those cells, the nature of how those cells change over time when they're injected into both small animal and large animal models. So the idea is that these are human embryonic stem cells that are then sent down a developmental fate, so that they have turned into an interneuron subtype that is very well characterized at the transcriptomic level, with the idea that these cells can then not go backwards, right? The feared complication that all of us, worry about is, does it go backwards and resume pluripotency? Does it turn into a teratoma? Does it turn into a different neuronal subtype that has an unintended effect? God forbid, an excitatory neuronal subtype. In my read of the preclinical data, I've seen no evidence of that. So I think that's very exciting, and looking from the outside in what's gone into preparing this cell type, it makes a lot of sense to me that this is a strategy that makes sense. Because if we don't have those assurances from the preclinical data, that these are cells that cannot in fact go backwards, then that's obviously concerning. The structure of this clinical trial, there's interval results now that have been released obviously in the public domain. But to summarize in short order, there's been a focus on patients that have mesial temporal sclerosis, like we talked about earlier, that globular, sclerotic, abnormal appearing hippocampus that oftentimes is smaller in volume and may have additional T2 flare signal there that we see in a significant subset of our mesial temporal lobe epilepsy patients. And the clinical trial involves making a very small incision on the back of the head, a very small burr hole or opening through the skull, and then using precise stereotactic techniques to deliver a cell delivery cannula to the hippocampus, and then a very slow, methodical, controlled set of cellular deposits so that we deliver those cells into the hippocampus. As it stands now, this is a cell therapy that is from another human being, is from a human embryonic stem cell line. So it does require immunosuppression. We don't know if these patients will need long-term immunosuppression. There's some sort of early signal that they may not, in fact, need that. So I think it's tremendously optimistic for the future that autologous stem cell therapies such as Aspen Neuroscience and others are working on that for Parkinson's disease, where the stem cells are actually made from a person's own germline or from other hematopoietic stem cells or adipocyte-directed stem cells that have then been taken into a neuronal lineage. I think that's a very exciting approach, because then it sort of takes away the immunosuppression part of things. So I think that there's a lot of innovation and science being done to make this out of the patient's own cells, and I think that's probably the future.